Thanks for joining us on another exciting journey into the world of science right here on the SANS Science Experience. If you enjoy this kind of content, hit like and subscribe. It's really the best way to support the channel and keep these coming.
Great to be here.
Today we're embarking on a deep dive into, well, the fascinating world inside our cells. Specifically, how the health of our mitochondria, those tiny powerhouses, profoundly shapes the identity and function of our key metabolic tissues.
Absolutely. It's quite amazing. We've been looking at some really cutting-edge research just published in Science, and it's revealing just how critical these little organelles are for our overall metabolic health.
Okay, let's unpack this then. Yeah. For you listening, our goal today is basically to understand how problems with something called mitochondrial quality control can trigger some really surprising transformations in our cells. Mm-hmm. Transformations that impact conditions like type 2 diabetes and liver disease. We're going to try and extract the essential insights, without getting bogged down in super technical jargon.
Right. Sort of the need to know stuff.
Exactly. Think of it as getting the cheat sheet to this important science.
Okay. Well, we already knew, or suspected, that mitochondrial damage is often seen in metabolic diseases. Things like type 2 diabetes, T2D, and metabolic dysfunction-associated steatotic liver disease. Used to be NAFD, now it's MASLD.
Right, MASLD. So damaged mitochondria were already on the radar for these conditions.
They were, but what this new research really illuminates is the intricate mechanism connecting that damage to the disease itself, how one leads to the other.
So it's not just that they're damaged because of the disease, but the damage might actually be driving the problem.
Precisely. And that brings us to this vital concept. Mitochondrial quality control.
Okay.
You can imagine it as like an internal affairs department just for your mitochondria.
Yeah.
Constantly working to keep them in top shape.
Internal affairs for mitochondria, like that. So what does that involve?
It involves managing their own genetic material. They have their own DNA, mitochondrial DNA,
right.
Maintaining their physical structure, which is quite dynamic, through processes called fission and fusion, splitting and merging. And crucially, a cellular recycling program called Mitophagy, which is specifically for removing damaged mitochondria.
Mitophagy. So it's like the cell is tidying up its power plants.
Yeah.
Getting rid of the broken ones.
That's a great analogy, yeah. And when we look specifically at T2D, type 2 diabetes, earlier observations had already shown that the pancreatic beta cells.
The insulin producers.
Exactly, the insulin producers. In people with T2D, those cells often show problems. Issues with their mitochondria structure, the activity of their mitochondrial genes, and their ability to actually perform that mitophagy effectively.
So in people with diabetes, their insulin-making cells seem to have power plants that aren't running well, right, and they aren't being cleaned up properly. That makes intuitive sense.
It does. And so the central hypothesis of this study we looked at was that these failures in mitochondrial quality control, they aren't just a side effect. They propose that these problems actually initiate a communication pathway. A signal goes from the mitochondria back to the cell's main control center, the nucleus.
Oh.
That's what we call retrograde signaling. Mitochondria talking back to the nucleus.
Retrograde signaling. So the mitochondria are sending out like a distress signal that actively changes the cell itself.
That's the idea. And they hypothesize that this communication is what actively drives the dysfunction, the failure of these crucial metabolic tissues.
Okay, this is where it gets really interesting. What did they actually find? Did this hypothesis hold up?
It did, very strongly. Their key finding is that when this mitochondrial quality control system falters, it activates what they call a retrograde signaling program.
Right.
And here's the kicker. This program doesn't primarily work by just killing the cells, which you might expect with damage.
Oh.
Instead, it causes the cells to lose their specialized identity. They sort of revert to a more immature state, a process called dedifferentiation.
Dedifferentiation. Hang on. So the cells forget what they're supposed to do. Like a beta cell stops being a good beta cell.
Exactly. Think of skilled workers in a factory suddenly losing their specific training. They're still there, but they're much less effective at their specialized job.
Wow. Okay, so losing their specialized skills. How does that happen? What's this signaling program doing?
So the researchers pinpointed a critical signaling pathway involved here. It's called the mitochondrial integrated stress response, or mtISR for short.
mtISR.
This pathway seems to get triggered by, well, various problems with mitochondrial quality control. Doesn't seem to matter exactly what the problem is. Could be issues with mitophagy, could be damage to the mitochondrial DNA, maybe problems with mitochondrial fusion.
So different mitochondrial problems all converge on this mtISR pathway.
That seems to be the case. It's like a common alarm system.
And what happens inside the cell when this mtISR alarm goes off? What are the consequences?
Well, the mtISR triggers really significant changes, right down at a fundamental level. It actually alters how the cell's main DNA in the nucleus is organized.
How so?
It changes something called chromatin accessibility. You can imagine the DNA as this tightly packed instruction manual. The mtISR can make certain pages, certain genes easier or harder to access.
Okay, so it's controlling which instructions the cell can read easily.
Precisely. And this, in turn, changes which genes are active, which proteins are made, and the net result is the cell losing its mature, specialized characteristics.
Right, the dedifferentiation.
Yes. And they didn't just infer this, they actually tracked the lineage of cells over time in their models and confirmed this drift towards immaturity. They could literally watch cells lose their identity.
That's incredible. So unhealthy mitochondria can essentially reprogram the cell's identity through this mtISR pathway. That's quite a powerful realization.
It is, and crucially, they also figured out what about the mitochondrial problems was triggering this. It seemed to be impacts on the electron transport chain and oxidative phosphorylation system.
Ah, the ETC-OXPHOS. That's the core energy generating machinery. Right?
Yeah, exactly. It looks like the disruption of energy production is what then sets off the mtISR.
So a breakdown in making energy acts as the sort of the first domino to fall, triggering this whole cellular identity crisis.
That's the picture that emerged from their work, yes.
Yeah.
And what's particularly exciting, maybe the most exciting part, is that when they managed to block this retrograde signaling pathway, this mtISR, in animal models, it actually helped the pancreatic beta cells, the really important ones, regain some of their lost identity. And significantly, it improved blood sugar control after the mitochondrial damage had already happened.
Whoa, hang on. Blocking the signal helped restore the cells and function, even if the mitochondria were still damaged.
Exactly. It suggests the signaling pathway itself is a major part of the problem, not just a passive reporter of damage.
That sounds like a potential game changer. Could we maybe target this communication pathway to treat diabetes?
It certainly points in that direction. It seems like a very promising new avenue to explore.
And you mentioned this wasn't just the pancreas.
That's right. What's also significant is that these findings weren't isolated there. They observed similar effects, this mtISR activation and loss of cell identity, in other vital metabolic tissues, like liver cells, hepatocytes, and also brown fat cells, brown adipocytes.
So this isn't just a beta cell issue for insulin production. It looks like a more widespread mechanism affecting metabolic health across different tissues.
It does appear that way.
Okay, let's maybe dive into some of those specific examples. What was the concrete evidence they found, say, in the pancreatic beta cells? Can we get into a bit more detail there?
Sure. So when they examined human islets, those are the clusters of cells in the pancreas containing beta cells from donors with type 2 diabetes.
Right, from actual patients.
Yes. The researchers found reduced amounts of mitochondrial DNA, less genetic material in the mitochondria, and also lower activity of mitochondrial genes, specifically within the beta cells themselves.
Okay, so less mitochondrial horsepower, genetically speaking?
Kind of, yeah. And these same T2D beta cells also showed a reduced capacity to perform mitophagy, that essential cleanup process we talked about.
Right. Less energy production capability and a less efficient waste disposal system for their power plants. That definitely paints a clear picture of dysfunction.
It does. And then they moved to genetic studies in mice, where they could be more precise. They specifically impaired mitochondrial quality control in the beta cells.
How did they do that?
For instance, by removing a key protein needed for mitophagy called CLEC16A, or by removing another protein, TFAM, which is vital for maintaining mitochondrial DNA.
Okay, so targeted sabotage of the quality control system. What happened?
These mice developed glucose intolerance, a hallmark of prediabetes, and they had fewer functional beta cells, a reduced beta cell mass.
But did the cells die off?
Well, that's the interesting part. This reduction in beta cell mass wasn't due to increased cell death, nor was it due to the cells failing to multiply.
So they weren't dying more or dividing less.
Exactly. It strongly suggested this other mechanism, dedifferentiation, was the main culprit. The cells were still there, just not acting like proper beta cells anymore.
So the beta cells weren't being destroyed.
Yeah.
They were, in a sense, losing their purpose, forgetting their job description.
Precisely. And when they looked at the genes being expressed in these mouse models, what instructions the cells were reading,
Yeah.
they found the beta cells were losing the genetic markers that define them as mature insulin-producing cells. And at the same time, they were starting to express genes usually associated with more immature progenitor-like cells.
Wow.
They even started producing some proteins that you just don't normally find in mature beta cells.
It's almost like their cellular memory was being partially erased or overwritten.
That's a good way to put it. And to really nail this down, they used those lineage tracing experiments.
Right, you mentioned those.
Yeah.
Tracking individual cells.
Yes. They could label beta cells early on and then follow them over time. These experiments directly confirmed that some of the original beta cells were indeed losing their ability to produce insulin. They were changing their identity.
That's really compelling evidence for the dedifferentiation idea in beta cells. Now, what about the liver cells, the hepatocytes? Was it the same story?
A very similar pattern emerged, yes. They looked at mice where they specifically created problems in mitochondrial dynamics, that fission and fusion process, or problems with mitochondrial DNA, but only in the liver cells.
Okay, liver-specific mitochondrial issues.
Correct. And in those mice, they observed the same thing. Activation of the mtISR pathway and a loss of the characteristics that define mature liver cells. And this was accompanied by measurable signs of liver dysfunction.
So liver cells also become less specialized, less effective at their jobs when their mitochondria aren't being maintained properly.
It seems so. And finally, they looked at brown fat cells. Those are the ones crucial for burning energy, generating heat.
Right. Brown adipocytes.
For those, they actually analyzed some existing data from a different study. It was on mice with a genetic defect affecting a really key mitochondrial lipid called cardiolipin.
Okay.
And analyzing that data, they found, again, a downregulation of genes that give brown fat cells their specific identity, and alongside that, an activation of the ISR pathway.
So even in brown fat, the same link. Mitochondrial problem, ISR activation, loss of cell identity.
Exactly. It suggests a pretty fundamental connection across these key metabolic tissues.
It really seems like a consistent thread, doesn't it? Okay, so this integrated stress response, the ISR or mtISR, when triggered by mitochondria, it really seems to be the central alarm system here, the thing that translates mitochondrial problems into this loss of cell identity.
That's precisely how the researchers interpret its role, yes. It consistently seems to be the pathway that gets activated by these diverse mitochondrial issues.
In that experiment you mentioned earlier where they blocked the ISR, that seems crucial. What happened there again?
Right, that's where the therapeutic potential really starts to look concrete. They used a drug, an experimental drug called ISRIB. It's known to inhibit the integrated stress response.
ISRIB?
They gave this drug to the animal models that had mitochondrial damage, the ones showing beta cell problems. And it helped restore both the number of functional beta cells, the beta cell mass, and their maturity. The cells started looking and acting more like proper beta cells again.
Wow. And this was without directly fixing the underlying mitochondrial problems.
Correct. The drug doesn't fix mitochondria, it blocks the stress signal. This strongly indicates that the ISR pathway itself is a critical intermediary, a necessary step between the mitochondrial dysfunction and the resulting loss of cell identity.
So potentially, we could treat the consequences of bad mitochondria by targeting this communication pathway, this stress response, even if we can't immediately fix the mitochondria themselves. That sounds huge.
It's certainly a promising way to view it, yeah.
Yeah.
A different angle of attack.
Did they look into what specifically triggers the ISR? You mentioned energy production, but what about other kinds of mitochondrial stress?
They did explore that. They considered other potential triggers, things like an increase in damaging molecules called reactive oxygen species, ROS.
Right, oxidative stress.
Or maybe an accumulation of misfolded proteins within the mitochondria. Both are common stress signals. But their findings pointed more strongly towards defects in the ETC-OXPHOS system, those core energy-producing processes leading to an energy deficit within the cell, as being the primary trigger for activating the mtISR in this context.
So it really looks like it's the cell sensing and energy shortage coming from the mitochondria that sets off this whole chain reaction. ISR activation, altered gene expression, and then the dedifferentiation.
That appears to be the most likely scenario based on the evidence presented in this particular research, yes.
Okay, let's try and synthesize all of this then. Stepping back, what are the sort of the big picture implications here for how we understand metabolic health?
I think the fundamental takeaway, the real core message, is that maintaining the quality control of our mitochondria is absolutely essential, not just for energy, but for preserving the very identity and the maturity of our key metabolic tissues.
And it does this by preventing this harmful signaling.
Exactly. By keeping mitochondria healthy, the cell avoids activating this detrimental retrograde signaling pathway, the mtISR, which leads to that loss of identity, the dedifferentiation.
Okay. And looking ahead, what does this mean for, say, future research or even potential treatments?
Well, this research really strongly suggests that targeting this mitochondrial retrograde signaling pathway, the mtISR, could represent a completely novel strategy. A new way to think about preventing or treating diabetes and maybe other metabolic disorders too.
Also, different from current approaches.
Yeah, I mean, instead of solely focusing on, say, managing blood sugar levels directly or trying to improve insulin sensitivity in peripheral tissues, we might actually be able to target the fundamental identity and function of the crucial cells themselves, like the beta cells, by modulating this stress pathway.
That's a truly innovative perspective, isn't it?
Yeah.
It's almost suggesting that the health of our cells' tiny power plants dictates not just their energy output, but their very nature, their job description.
It certainly seems that way. And it also, I think, opens up this intriguing possibility. It challenges the maybe older view of mitochondria as just passive recipients of signals from the rest of the cell.
Right, just energy factories.
Yeah. This suggests they play a much more active role in actually shaping the characteristics, the phenotype of our tissues through this kind of communication.
So this research really shifts our understanding of the dynamic interplay within our cells. It's not a one-way street. What unanswered questions jump out? Where does the research go next?
Oh, there are many exciting avenues. I mean, future research will undoubtedly focus on dissecting the precise molecular mechanisms. How exactly does the ISR alter the organization of DNA, the chromatin accessibility?
Right, the nuts and bolts.
Exactly. And how do the different sensors within the ISR pathway actually detect these different kinds of mitochondrial problems? Which sensors respond to energy deficit versus, say, protein folding issues? There's still a real wealth of knowledge to uncover about this intricate communication network.
Absolutely fascinating. It just underscores how interconnected and incredibly dynamic all these processes within us truly are. It's not simple plumbing.
Indeed. No, this research really offers a significant leap forward, I think, in our understanding of this complex relationship between our mitochondria and the identity, the fate of our metabolic tissues.
Well, this has been incredibly insightful. Thanks for breaking that down for us.
My pleasure. It's exciting work.
Thanks for listening. And as always, don't forget to like the video and subscribe if you haven't already. And maybe take a moment to think about how the tiny powerhouses within your cells are not just fueling your body, but actively maintaining the very nature of your tissues. What other surprising roles might these crucial organelles be playing that we haven't even discovered yet?